Hi, thank you for the introduction. So as mentioned, I will be looking at the sex differences in resting state CBR. The high prevalence of physical inactivity is of concern to the development of many diseases. Physical inactivity alone has been attributed to 5 million deaths globally. Has been shown to increase the risk of dementia by 40%. Furthermore, physical inactivity has been shown to increase by 24% the increase to the development of cardiovascular diseases and to a high incidence to the development of obesity. As we can notice from the figure above from the darker areas, the prevalence of physical inactivity in males is quite high. In females, the prevalence of physical inactivity is even higher. Fortunately, physical activity can help prevent and manage many of the diseases. The benefits of physical activity has been shown in both vascular and structural changes. It has been shown as well to increase cerebral blood flow, gray matter volume, and brain-derived neurotrophic factors, which further increase neuroplasticity and neurogenesis. Physical activity has been shown as well to increase white matter organization and decrease oxidative stress. However, although we are aware of all these positive changes that physical activity brings to these physiological quantitative measures, we are still unaware of the optimal dose and intensity of physical activity. The World Health Organization Physical Activity Guidelines has proposed a minimum of 150 minutes per week of moderate to high intensity physical activity. If we take a closer look at the guidelines, you can notice that there are no sex differences within the implementation of these guidelines, with the exception of postpartum and pregnant females. The generalization of these guidelines may not be optimal to the benefits of physical activity for males and females, as this research has been based on an overrepresentation of young white males, and there's also no regards with its potential benefits on brain health, and even more specifically, vascular brain health within these guidelines. But it has been shown that there are positive effects to physical activity on vascular brain health. As noted by Thomas and colleagues, they showed that in older master athletes, they had a greater relative cerebral blood flow compared to sedentary older individuals and young controls. Now, as presented earlier, the CBR and CBF are two of the most important biomarkers of vascular brain health. So from this study by Liu and colleagues, we did notice that from the decline in through aging, the decline in vascular brain health, CBR appears somewhat to give us more of an insight, as we've discussed earlier, with the potential compensatory effect of CBF. So therefore, for the purpose of this study, we decided to use CBR to analyze these effects of physical activity on vascular brain health. In 2017, Liu and colleagues developed a new magnetic resonance imaging technique known as resting state CBR. What is resting state CBR? It's simply a measure of CBR, but obtained through the resting state fMRI bold data without any respiratory manipulations. As we've discussed yesterday, the lower frequencies of the bold signal hold valuable physiological information, including a proxy of the end tidal 2 as we apply a low-pass filter. However, these lower frequencies do not hold any information with regards to the millimeters of mercury of the end tidal CO2. Therefore, from this technique, we obtain relative CBR, which will be discussed further in this presentation. The main advantage of this technique relies upon its potential implementation within many large data sets, such as the UK Biobank, the HEP and ADNI data sets, as they only offer resting state fMRI bold data. The figure here illustrates the resting state CBR pipeline starting from the pre-processed bold. Following smoothing of the bold data, we conducted time domain filtering. The blue signal represents the original pre-processed bold signal, while the red signal represents the filtered bold signal. As we can notice from the red signal, it resembles natural fluctuations within the end tidal CO2. The filtered bold signal is then used as a regressor within the general linear model. The bold variable is simply replaced by the original preprocessed data. And from the computation of the general linear model, we obtain the CVR index as written in red. Then further from this, the CVR indices are normalized by a whole brain average of the CVR, which generates this relative CVR map. Previous work in our laboratory has shown a complex interaction when looking at the effects of physical activity, BMI, and brain structure. And even more importantly, they have shown significant sex differences across all these variables. Therefore, for the purpose of our study, we decided to include as a secondary objective BMI when looking at the effects of vascular brain health. From the objectives of the study, the primary objective was to evaluate the effects of intensity and dose of physical activity on CVR in both males and females. The secondary objective was to assess the effects of BMI as a measure of obesity on CVR in both males and females. So the human connectome project aging dataset was used for this study. It includes 234 males and 297 females older than 36 years. The resting state fMRI data was already minimally pre-processed with motion correction, field map correction, linearity trending, and registration normalization to MNI space. And from the International Physical Activity Questionnaire, the IFAQ from the HCP dataset was used to quantify physical activity intensity and dose. So more onto the quantification of physical activity intensities, it was categorized into three groups: light, moderate, and vigorous. So from the IFAQ, we converted the intensities and duration into walking metabolic equivalents of tasks known as METs. The dose was simply a combination of all intensities in METs. So to ensure the validity of resting state CVR within our own data, we decided to analyze the differences between resting state CVR and hypercapnia CVR using arterial spin labeling in 7 healthy participants from the Leska laboratory at the Montreal Heart Institute. So as we can notice from the two figures, there appear to be some differences within the sensitivity, but the overall representation of CVR remains quite similar. Using a Pearson correlation test, we noticed a very high correlation between the resting state CVR and hypercapnia CVR with an R value of 0.96. This provided us with the confidence to use resting state CVR in assessing the effects of physical activity on vascular brain health. Now, coming back to our first objective, now looking at the effects of physical activity on CVR in females, we noticed through the frontal cortex positive linear relationship when looking at moderate to vigorous, and total physical activity. Interestingly, when looking at the temporal cortex, we notice negative linear relationships at higher intensities with vigorous and moderate to vigorous physical activity. In males, we only notice negative linear relationship within the frontal cortex with low and total physical activity. From the second objective, looking at the effects of BMI on CVR in females, we noticed a positive linear relationship within the whole gray matter. Males, we noticed the opposite effect within the temporal cortex, having a negative linear relationship with BMI and CVR. It is to be noted that all these analyses, especially within the first objective, were multiple regressions, including systolic blood pressure, BMI, and age. So similar to our results we obtained in females within the temporal cortex, Inside and colleagues showed a negative relationship between CVR and cardiorespiratory fitness, primarily in the temporal and parietal cortices in older adults. More recently, Dubose and colleagues showed a nonlinear relationship to the effects of maximal aerobic capacity and CVR. However, these analyses did not conduct sex disaggregation analysis. Now, looking on the lifetime effect of physical activity, Thomas and colleagues showed that in master athletes, across the whole brain, they had a lower CVR when compared to sedentary individuals. And looking back again at the temporal cortex, they had for a lower, within the temporal cortex, a reduced CVR for this higher aerobic capacity. So the results are confusing. As it's been attributed that lower CVR in disease state is associated with poor vascular health, these implications with regards to physical activity may not be interpreted the same way. There's a potential effect of chemosensitivity as proposed by Thomas and colleagues, where CO2 chemosensitivity attenuation in adults with a high exposure to aerobic activities may explain some of this lowering of the CVR within fitter individuals. Now, looking at cerebral autoregulation as proposed by Insan and colleagues, a reduction of this blood pressure reactivity through the aerobic training may diminish the response to the physiological stimuli, which would overall decrease this onset of cerebral autoregulation, here again, potentially lowering that CVR. Coming back to the secondary objective, looking at the impact of BMI on vascular brain health, it has been established that by Knight and colleagues that a reduction in cerebral blood flow is associated with higher BMI. However, when they noticed a complex interplay when looking at physical activity intensity in relation with BMI and cerebral blood flow. Insan and colleagues, as shown earlier, were the first to show sex differences in the relationship between BMI and gray matter volume. So from these results and our findings, we suggest that the impact of BMI on vascular brain health seem to be influenced at least by sex-specific mechanisms. To conclude, these findings suggest sex differences in the effects of dose and intensity of physical activity and BMI on CVR. Future studies should explore the potential physiological adaptation in fitter individuals that may affect these CVR values. Would like to acknowledge all members of the Quantitative Physiology Imaging Laboratory, as well as all collaborators and funding support. Thank you.